A neuron transmits nerve impulses through itself, but when it's time to send a message to another cell, most neurons will send chemicals to the next neuron across a very small gap between them. The junctions between neurons and the next cell are called synapses. There are three major types of cells that neurons will form synapses with: sensory receptor cells, other neurons, and effector cells. A sensory cell could be part of the eye, ear, nose, skin, or any place where senses are used. This sensory cell is connected to the skin and will pick up pain sensations. The pain message may be sent to another neuron that could be part of the spinal cord or brain. These intermediate neurons are also called interneurons. The message can then be sent to a cell that will have an effect on another cell, so they're called effector cells. In this example, the effector cell is going to signal a muscle cell, which would move this area of skin away from the source of pain. These three cell types together make a simple reflex arc, going from sensation to the central nervous system to an action. The chemicals released into the synapse are called neurotransmitters. Many neurons have a very small fluid-filled gap between the presynaptic cell and the postsynaptic cell. This gap is called the synaptic cleft, and it's only about 20 nanometers wide. It's a really, really tiny gap. Now that we know some of the basics, we can look at the steps of synaptic transmission in more detail. First, a nerve impulse in the presynaptic neuron reaches the end of the membrane. This causes calcium ions to diffuse into the cell through channels in the membrane. The influx of calcium causes vesicles with neurotransmitters in them to move and fuse to the cell membrane. The neurotransmitters are released into the synaptic cleft by exocytosis. They diffuse across the synaptic cleft and bind to receptors on the postsynaptic membrane. The binding of neurotransmitters triggers sodium ion channels to open and pass the threshold potential. The action potential is propagated along the postsynaptic neuron. Then the neurotransmitters are broken down and removed from the synaptic cleft. If there isn't enough neurotransmitter binding to receptors, then there won't be enough sodium flowing into the cell to reach threshold potential. This means that there is an all or nothing response. Either there is enough neurotransmitter to start the action potential and the impulse is sent, or there is no impulse. In this